So here in introduction we can see here, right? It is the process of multiplication in which a portion of fragment of the plant body functions as a propagate and develops into a new individual. When we are cultivating, we are taking any portion. It may be the seed, it may be the plant part, or it may be the some while doing micropropagation, we are trying to generate or develop a new plant which will have all the features or quality which is there into the proper origin. It is also known as asexual propagation. When it is vegetative propagation, it is known as I told you, three types of propagation: seed propagation, vegetative propagation, and a micro propagation. Right. So seed propagation we discussed in the last class, like boat casting method, dibbling method, and the nursery beets method. Right. So here there are different types of vegetative propagation. The types are natural, artificial, cell culture followed by differentiation, aseptic or micropropagation. It is also known as clonal propagation, and by fermentation with the help of different types of microorganisms. Right. So let us discuss the first one, natural methods. Natural methods means vegetative organs, for example, roots, stems, leaves, rhizomes, tubers, and bulbs. These are the part of the main plant we are taking to generate the new plant, right? So that's what the God has given the plant in a such a way, the features, that only one part, any of the part it may be, having the totipotency capacity. So what is totipotency? It is the part of the plant which is having capability to generate new plant. That is called a totipotency. You might have discussed in your inter classes in botany, right? So that quality, which part is having in the main plant, that we need to identify, and with that we are going to initiate our cultivation. Here there are different examples given. The first one, bulbs. So which plant the bulbs is having capability to generate new plant? Examples are garlic, right, and squill. Squill is the glycoside, cardiac glycoside, which we discussed in the classification of crude drug example in glycoside. So these two plant bulb we can take to generate the new plant. That is the natural method. In second example, it has given tuber. So tubers, in the tubers jalap is there and a potato. You might have seen into the village that farmers, when they will cultivate, what is the type of potatoes tubers they are taking, because potato itself, it is comes under the tubers category that we have seen in morphological classification. So that tubers of the potato, the potato itself is known as tuber. So that potato we are taking and we are placing into the plug the soil at in a specific space, and they are growing into the plant. The third example, that is rhizomes. In rhizomes there are two example given, that is the ginger rhizome and turmeric rhizome. So ginger, whatever the ginger we are using, even for preparation of tea and preparation of different masalas, right, that is the part itself, it is known as rhizome. So that ginger we can take it as a initiator of our medicinal plant. Even in case of turmeric also the same. The next one is the suckers. It is licorice. Licorice suckers we can take for generate a new plant. And runners, the mint or pudina or chamomile, these two plant is the example for the runners. These are the natural method. And when it comes to the next one, artificial methods, we are going to, it is the specific method or process is used on any vegetative part of the plant for propagation, right? For example, here five methods has been given in artificial method. The first one, cutting. As the name indicate, cutting. We are going to cut the medicinal plant and we are generating the new plant. Layering. Layers we have to make into the plant and we are going to generate. Division. The cells we have to separate and divide in different division, and then grafting techniques, it is very popular. You might have seen in the case of rose or in the lemon, citrus species, right? And budding. So let us discuss what is the type of different types in artificial method that we are going to have. The next one, first one, we will discuss the first technique, that is called cutting. A cutting is separated portion of root, stem, or leaf, right? We have to cut it from the parent plant root if it is possible. We have to we will be having the reference that in this particular plant which part is having that totipotency capability, so that we can select that particular part of the plant and we are going to cut it. It is very common method used for artificial vegetative propagation. For example, sugarcane. All of you know that sugarcane juice you are having. So sugarcane, when we are going to uh, cultivate, we need to take cut it the particular rhizomes, or in the case of stem, uh, and a rose, the in different varieties, different colors of rose are available, where we are going to adopt the cutting technique. In citrus plants like lemon, orange, or so many other types of oranges are also available, that we are going to employ the cutting technique. Then duranta, grapes, coca, and bougainvillea, these are the different examples of plant when we have to cut the portion and that we are going to employ. Let us discuss about the different types of cutting, how we are going to cut and we are going to cultivate the medicinal plants. So here four types given. The first one, stem cutting. The second one, root cutting. Then third, tubers and offsets, right? So one by one we will discuss. The first one, stem cutting. Single node stem cutting. If internode is greater than 1.5 centimeter, right? So here the stem size is matters, right? So if the internode, the main portion of the particular plant, if it is more than 1.5 centimeter, we have to go for single node stem cutting. If it is double node in stem cutting, what happened here? The internode is only 1.5 centimeter. Then we are going to go for double node. Multiple node stem cutting, if internode is very small, then we have to cover the main portion, because in stem, if you see the transverse section of the stem, there are a lot of part: cortex, cambium, stone cell, starch cell, xylem, phloem, fibers, pith, right? So whenever we are taking the sample for going for the cultivation, the important cells part supposed to be there in your sample, so that it will be able to generate the plant. If few cells are missing, and if cells are not there in the sufficient quantity, they will not be able to germinate, right? Soft wood cutting and hard wood cutting, right? So the wood also, we need to understand that soft wood cutting it is required, or hard wood cutting it is required. It vary from plant to plant. Here you can see the soft wood cutting. Example given, the berry, right? Here how they are cutting the wood here uh, in this particular plant. The wood, it is very soft. Okay, so in berry plant we need to cut the slowly into this area, so that the wood portion supposed to be there whenever you are going to cultivate this particular plant. And then this we are going to uh, put into the soil wherever it is available, if it is possible in pot or in the open land or in the nursery beds where you want to cultivate. Then you have to dig into the we need to make some holes into the soil, and that we are going to insert, so that they will be able to develop the roots and further growth. Here you can see, this is the another example. Here in the case of rose, in the case of al- almond, or in the case of orange, how we are going to cut the woods, right? The next one, the root cutting. In the root cutting, the brahmi, right? Brahmi is the example. The propagation with root cutting, only roots are required. If you will uh, cut the root from the parent plant, which will cover the maximum portion of the root for the further development, and if you will put into the different pot or different place wherever the proper nutritional requirement of that particular plant it is fulfilling, it will able to develop the leaves and stem. Here you can see how they cutted the root part, this part from here. If you'll cut this particular area, and this root if you will put into the another pot, how it is able to generate the stem and leaf. Here the green part it is able to generate. In the case of brahmi, you can do this technique. The tubers, the examples are potato, already we discussed, and the yam, right? So in this you can see here in the yam how they are tubers, they have cut it into the different pieces. And this, if you will put into the proper soil, use of ice in potato tubers. So in, if you see the potato properly, there will be the this kinds of structure, the texture of the potato surface, it will be there in like this only. So whatever the if you are cutting this potato in the many pieces, here you they have how you are going to cut, they have given the sample here. So in each cutting, at least one eye in the sense, it will be the structure, the texture it is like that, so it will look like a eye. So here you can see in this potato cutting there is a one eye, and in this potato cutting two eyes are there. Here also two eyes, here also two, here one. So this eye, it is having capability to develop the plant or leaves, stem, and further development of the plant. So the cutting should be there in that way. If entire potato if you are putting also, the one eye will be start their functioning and it will develop the plant. So likewise you have to take care when you are going for the tuber. In case of offset, so here in the case of asparagus or in the case of banana, two example they have given, banana and asparagus. So here you can see here in this how they are cutting the offset. This is called a offset, the set which is there I can see in offside, right? So here this portion, in the case of what do you call Rax Bargiana, or in aloe vera also it is possible, which will be this proper outside. If you will remove it from the main plant, and if you will go for, so the root pocket it will also cover, and it will start generating the new plant. So in banana also you can see how they are doing banana. That is the very good example. The second example of the second method of the cultivation, that is the layering, right? It is in layering method one, we can see here the propagation by layering. Here in the case of guava, mango, lemon, and jasmine, we can adopt this particular method. Here in the plant, you can see this plant it has gone, and if you will cut this particular area, and if the from here this area, it will having the uh, capability to generate the new plant, right? So here the layer of the plant can be used for the generation of new plant. In method 2, the stem is uh, grilled to introduce root formation above the cut. Here you can see the grilled stem is covered with damp moss. Here they are grilled, or they removed the stem, only wood part it is there. And this you have to covered with the damp moss or some other types of moss where it is from another plant. Okay. And that you have to cover properly. Aluminum foil or plastic setting is wrapped around the moss and tied at both end. You have to tie here so that it will be covered properly. This cover is removed 2 to 3 months after tying, or when the roots can be seen, right? So if you do so, the root will start growing from this side. And if you feel that root is developed already by touching this, or by from this hole, if roots are coming out, later on after 2 to 3 months you can remove this pocket, and it can be generated as a new plant. So this is the second method of layering. Here you can see how in the real image how they have to remove the stem part, the main cortex and cork texture of the stem, that we have to remove till the wood. In the case of cascara bark, lychee fruit is there. In citrus, lemon, orange. In the case of pomegranate, right, and guava and croton. So these are the different examples for layering method 2. The next one, the third one, it is division, right? Division means we have to separate, as the name indicate, division. We have to separate it from their group. That in the case of rhubarb and gentian root, dahlia, and male fern. In this, we have to divide it, and we can place it into the different place, so that they will be able to generate, or they will be able to grow into the new plant. The fourth method, that is known as grafting techniques. So in grafting techniques, there are two types: approach grafting and true grafting, or it is also known as stem grafting. The term stock the rooted plant, or graft, or seon, here detach the living portion of the plant, right? So rooted plant and the detached living portion means we have to take the another branch of the another plant, and we have to graft it into the main plant, host plant. Approach grafting, here in approach grafting you can see the root stock is there and the section is there, which we have cut it out. So in the root stock, we have to join together in a such a way, so that their inner cell should come into the contact of each other. Here the the proper grafting it is make in a such a way, you can see here, right? So when we are joining them, this inner portion and this inner portion, they will join together. We have to tie it properly here. So whatever the season we have done here, that will be the with root stock, it will start growing into the new plant, which will be having the feature of both the plant which we have taken. Here you can see in approach graft one plant and another plant, both we have to cut in a such a way, so that the inner cells, here you can see cut 1 and cut 2. This is the stalk and this is the season. So they have we have to tie it properly. Here you can see here in the image how they have cut it. This one, after cutting we need to tie it, then both after few days this both they will be joined together, and they will start giving the nutrient to each other, and whatever the cell development here occur, that will be having the quality of both the plant, stock and season plant, and you can get the hybrid quality of the constituents. True grafting or stem grafting. In this method, only stem are involved for grafting. So two types are there: whip grafting and cron grafting. Let us see the what is whip grafting. Stem with same thickness are selected for grafting, like apple and watermelon. So whenever we are selecting, they should have the same thickness. Why same thickness is required? Because ultimately they are going to join together. If one thickness will be more, for example, here in A and B has been given. This is the A stem, this is the B stem. So we have to cut the stem in a such a way so that it can be inserted here and it can be tied. You can see here in the C image, this zigzag shape of the cutting, here they have joined in this hole and they have tied it. Now this is the different plant, this is the different plant. Now they have grafted. So once grafted will done, when whatever the plants it will grow, it will be having the quality of the both the plant, and you will get the uh, good quality of the apple fruit and watermelon fruit. When it comes to the crown grafting, this is the second type of grafting in stem grafting. Seasons are inserted into stalk like Maristica fragrance and Cinchona bark. In these two medicinal plant, this method it is suitable. Stalk means the stalk root which is there, that we need to insert. We need to make the hole in a such a way like crown. We have to prepare like that's what it is known as crown grafting. Okay, then we have to tie it properly. So again they will start generating the new type of plant. The next method, the fifth one, that is called budding. Budding means in the case of sweet orange or in the case of ornamental plants or in the rose, we need to cut the portion from the stem and that we have to insert in another plant that will have the capability, the totipotency capacity, which the part of the plant having to generate the new plant. So that we have to insert in that main plant in a such a way so that it will be stick properly, and the interchanges of nutrient materials and development of the cell will be there in a such a way so that it will have the capability to generate the new plant. Okay, so this method also it will be done. You can see here in the image how they are cutting the outer portion, the bark portion, in such a way. Here in the first image you can see that they are cutting here, and this portion they have to cut in such a way so that this particular portion which you have cutted from one plant, that can be inserted here without exposing the extra part. The exposure should not be there unnecessary. Whatever the inner portion of the first plant and the inner portion of the second plant, both they join together in a such a way so that whatever the plant will come out, it will be having both the quality, and that's what we are going to get, the hybrid quality, right? It is not simply it will come if you'll insert the increase the giving plant growth regulators or different types of chemicals, you are not going to get. So this technique we need to adopt. Cell culture followed by the differentiation. How we have to do this? It is a collection of technique used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture medium of known composition. So, what do you mean by known composition? We need to understand scientifically that the plant or cells which we are taking to generate the new plant, what is its requirement? Means macroelement, macronutrient, micronutrients, what carbon source, so what is the type of nutrient that particular cell required. We will be having the reference data as per the monograph that we have to understand, and we have to prepare that culture media in a such a way so that it will fulfill the requirement, the nutritional requirement of your sample tissue or sample cell. Okay. And here one word used, you can see here in the third line, under sterile conditions. What do you mean by sterile conditions? It should have, it should not have any types of microorganisms, right? Because if microbes will be there, they will start utilizing the nutrient materials, and your plant will not get those nutrient which you have given for the plant. That's what we need to do the sterilization techniques. That lot of sterilization techniques are there, right? That you might have studying in the microbiology, like different hot air, sorry, this dry heat sterilization, moist heat sterilization, right? Ultrafiltration, though different types of, so depend on, suppose if it is liquid sample, the nutrient medium it is liquid. So for liquid, what is the type of sterilization? We need to moist heat, right? Autoclave is there. So 121 centigrade temperature and 15 lb pressure, 15 minutes depend on the volume of the culture. We have to go for sterilization. We are making the medium free from microorganisms. Then that is the food, that is nothing but food of the plant. That we are different types of techniques it is available in that, like conical flask, petri dish, or different other glasswares, where we are going to give this nutrient to the your plant which you have taken as a sample. It is widely used to produce clones of plant in the method known as micropropagation. That's what it is known as, micropropagation. So here you can see the schematic diagram of the micropropagation. First, we have to harvest the cell. We have to identify what plant you are going to cultivate, and in that plant, which part is suitable to generate the new plant. Same thing what we have done into the different techniques here in the grafting techniques and budding layers. Like that, we need to select one part, right? By understanding scientifically, then isolate cells with the use of appropriate enzymes, right? So we need to isolate, like different isolation techniques are available, centrifugation we have to do. Then we need to prepare the cell suspension. Then whatever the sample you are going to take, in that maximum number of viable cells supposed to be there, right? Living cells, I mean to say, the cells are in the living condition, and that they should able to divide themselves, right? Or multiplication, that is important here. Apply the isolated cells onto an appropriate growth media in a culture dish. So here you can see small petri dishes are available. In that, you need to apply this isolated cell. Which isolated cell? Which you have harvested from the plant and you have isolated or separated from other cell debris. Means in that sample, cells only supposed to be there. Unwanted materials like fibers will be there, and some cell debris which is not having any totipotency capability. That why you are going to give unnecessary, because ultimately when it will grow as a plant, automatically they will be able to develop all these materials which is required for their normal growth, right? The culture cells by placing the culture dish in a cell incubator. So we need to incubate. The cells are placing in the culture dish. After you have putting into the petri dish, you need to put into the incubator. Incubator is what? it is the one kind of chamber, right, where we are going to maintain the temperature, so that how much temperature it is required, your plant cells, if it is required more temperature, it is required a low temperature, what kind of temperature it is required, that we can maintain as per the requirement of the plant. Subculture cells to obtain a pure culture or bypass some problems, for example senescence or some other types of, if it is required we can remove that unwanted part and we can transfer. Verify the cultured cells are of cell type of inserted, right? So what is the type cell types it is there, then at last cells are ready to be manipulated or modified for a experimental procedure, right? So this we have to do step by step. So here two thing only you need to remember: cell and nutrient. We have to take one sample cell and one it to start, right? So here the next one, the micro propagation. Coming to the micro propagation, it is a practice of rapidly multiplying stock plant material to produce a large number of progeny plants using modern plant tissue culture methods and all those things. Can get high yield of short period of time, and micro quantity of explant is enough to produce the many plants. So explant, what do you mean by explant here? Explant is a sample, nothing but a sample, micro quantity. We need not to take the big branch or big tree. It's not like that. Which plant, which particular part it is having capability to generate the new plant, that micro quantity we are going to take, and that is enough to produce the many plant. It is not that from one explant only you can generate only one plant. You can generate the in number of plant. So let us discuss the procedure, micropropagation procedure. Selection of an explant from a mother plant that is healthy and vigorous. Unusually, apical buds are preferred as explants, but any other tissue can be used. And the establishment of this explant in a plant culture medium. A medium support growth and cell division. Depending on the plant requirement, different types of media are used for a specific type of plant. This already we discussed. So multiplication. What do you mean by multiplication? In this stage, the X plants gives rise to a callus, right? In callus culture also we have to prepare the callus solution. So here I need not to go deep into the plant tissue culture. You need not to have only just a method name, and what is the method, that only you should know. Differentiation and organogenesis. So these two terminology it is you are supposed to write. So here steps involved in vitro micropropagation. In vitro means in the laboratory with the help of glasswares and micronutrient which we are going to do, that is called in vitro. So cleaning of glassware, first step you can see here. We need to whatever the conical flask, beaker, petri dishes, test tubes, whatever the glassware we are going to use for micropropagation in the laboratory, that we need to clean properly. Here cleaning in the sense it is not just a washing, it should be free from microorganism or sterilize, I can say sterilization. How now you're sanitizing your hands, sanitizing your bag, sanitizing many things which you are using in your daily life, right? So now no need to explain that sanitization how to do. You are very much aware nowadays. So definitely when you are working into the laboratory for the plant tissue culture, you need to, first of all, the lab should be aseptic condition. Aseptic condition means there should not be the microorganism like HEPA filter and double door techniques, disinfection of the legs, wearing of the gown or apron, gloves, mask, all those things it is required. Then second step, the preparation of nutrient medium. We need to prepare the media like sodium, potassium, zinc, iron, carbon, calcium carbonate, nitrogen source, carbon source, activated charcoal. What are the plant food we need to understand, right? So that food we have to prepare, like how you are preparing biryani, pizza, burger, and all those things for you people. Like that, we need to understand what is the plant requirement. That requirement we need to understand, and we have to prepare the plant food. Then the selection and sterilization of explant, right? Selection and sterilization means which explant is nothing but I told you sample, right? So in this sample, uh, we need to select which part, like we have discussed in the different, like some people, some plants, they are having bulb is having totipotency, some plant roots are there, some plants rhizomes are there, the capability, so some plant leaves, bark. So which part it is suitable to generate the new plant, that we need to understand, and we have to select. Here the selection means the selection of the part, then sterilization of explant. It is not that simply you selected the root, you some from somewhere you have cut it out the root and taken it from the uh, field, and you start generating. You need to wash it properly by the tapped water, then distill water, then double distill water, then triple distill water. See, four types of washing only I told. So washing is not the part of sterilization. Here you are removing the earthy matters, sand particles, foreign organic matters, microorganisms, insects, or some unwanted thing which is there with your explant, right? Then you have to go for sterilization. So if I say sterilization, sterilization is the process to making the thing free from microorganism. So free from microorganism is the sense how you are going to make it free from microorganism, because these are the plant. If it is glassware, you can put into the oven, hot air oven, and at 180 degree temperature, you fuel heat that, all the microbes will kill because of high temperature. But if you do so with this plant material, what will happen? You will get the ash. It will burn completely, and there will be no living cell. It will become a roast, like chicken roast or something like that. Okay, so that's what. There are different sterilization techniques for the explants. If it is leaf means there is no question of heating. We cannot heat the leaves. We have to go for surface sterilization. There are different chemicals available, like sodium hypochlorite, alcohol, 70% of alcohol, right? Sodium nitrite, right? All those chemicals, we have to take the cotton, we have to dip, and we have to wipe the surface of leaf. You have to wipe the surface of bark, we have to wipe the surface of root, right? So likewise we have surface sterilization. And if it is we sometimes we are completely immersing those plant material into the disinfectant liquid, or sometimes if it is required, we are putting into the different lamp, UV lamp, or right, radiation, that is called like ultrafiltration. With the help of different radiation, we are trying to kill the microorganisms without killing the living cells into the explant. The fourth step, that is the incubation of aseptic explant into the nutrient medium, right? So means inoculation, sorry, inoculation of aseptic explant, right? Inoculation means introduction or putting into the aseptic explant into the nutrient medium. Now we are mixing the plant and the medium. This is nutrient medium is food, and aseptic explant is plant material. Both we have to inoculate, or we have to introduce. Both we are mixing together. Nothing, inoculation is nothing but introduction or mixing, right? Then next, the fifth step, that is the proliferation of shoots on a multiplication medium, right? So we need to, these shoots, whatever we have assembled, that we need to put into the multiplication medium where they are going to multiply like any what do you call conical flask or petri dish or some other types of pot where the plant is going to transfer the shoots for the subculturing. So before going to the culturing, it has to go for subculturing. So in that, we need to transfer the shoot into the subculturing media. Then next step, rooting and hardening up plantlets. So we are allowing it for few times so that the root can develop and this soft medium and the explant they will become hard, right? Then we are going for field trials, or this particular developed plant we can go for transplant into the open field so that they can further develop into the new plant, right? So likewise we are going to do the micropropagation. Here you can see into the image for the better clearance, tissue sample from any region an adult plant is cultured. Here you can see they how they are taking the leaf part. Here in the leaf the different epithelial cells are visible here. So this leaf cutting part from one plant we need to take as a tissue sample, and here in this petri dish there is a nutrient medium, and this leaf part we are keeping here, and we are allowing it for uh, undifferentiated callus form. So it will start growing here in the callus form. Then we again we are transferring it to the callus separated and single cell cultured, right? We need to separate it, and then we have to go for single cell culture into the medium into the conical flask. Then we are transferring into the one test tube, right? The further culturing generates a new plant, then it will start developing of the root. You can see here roots are developed, and the yeah, at the same time leaves and other stems are also growing out, right? Then it will go for the what do you call next plant. Here the auxin cytokinin ratio regulates the uh, morphogenesis of cultured tissue. Morphogenesis means the development of the cells. The morphology of the plant will start growing. Leaves will grow, stem will go, root will also go inside, right? So here agar nutrient they have given as the example. So agar nutrient in the plant tissue we have to keep into the test tube. This is the plant tissue, this is the nutrient medium. Here the ratio, the oxygen and oxygen, here they have taken the kinetin, that is the one of the hormones, right? Here the callus is developing in this this was small, and here you can see it is developing in more, and the root development also you can see in downside, right? And here the shoot development, and here the no growth. So here they have given the ratio of auxin and cytokinin, how much ratio you are taking, right? Mg universe here the here the high auxin cytokinin ratio root development it will encourage. If low auxin and cytokinin ratio, it will encourage the shoot development, or intermediate level it will develop the callus. So here you can see different 3 is to 0. 0.2, 3 is to 0.02, and 0.003 is to 1. So different ratio. Here ratio is 15, 150, and 0.03. So different ratio if you are taking, you can see the different types of growth into the plant. So that's what we need to very much particular about the ratio of the auxin and cytokinin, how much we are giving for the morphogenesis expectation. Here you can see these are the real image of which has been done into the lab, right? So here 4 5 types of what we have discussed in just now schematic diagram, that is only. Here the small callus is there. Here you can see the callus development. Here you can see the root development, and how it is going as the plant development. Here also you plant this we can perform into the lab once you will come to the college. Just for seeing this, you can see how the plants are. This A image, this is the B image, how the plants are developing in that small test tubes, and with different ratio of plant growth regulators, we can see the different types of development. And this is for different 4 test tube. In D image, this is small one, this is big one, This is a small one, right? So likewise it will go on. Okay, here you can see in this image also how it is going to develop. Okay, so next we'll discuss in the next class further. It will be in continuing. Okay. Hello everyone.